All right, good evening, all. We'll start today's session. This week's session actually, most of the concepts you guys might have already learned in high school. So, like Bacillus thuringiensis and all those stuff. So, we'll start with the questions and then follow up along with the concepts. Okay, like previous tutorial sessions were mostly like first we kind of revise the week's lecture content and then we look at the questions. So this tutorial session will have the questions along with the concepts. Okay, so I will start sharing. So actually the first question is sample question is um, eradication of boll weevil was achieved by the use of which particular technique? Now, boll weevil are insects which feed on cotton bolls. Okay, so it was actually a huge um, kind of an epidemic kind of thing, especially when it started in the US. So I will just walk you through this video just to give you an idea that how big a problem was boll weevil and why was its eradication like program started. Okay. So hopefully the sound, don't know if it's audible, but the captions are on as well. You can hear, I mean, or read. So basically, this was such a huge problem that one used actually biotechnology to genetically maneuver these boll weevil, these pests. And so what is it that they did? So the YouTube video that I showed, the link is given here. If you wish to watch it properly with a proper volume, you can watch it. So basically what one does is one creates sterile male insects. Okay. So the mechanism of reducing the number of insects in the affected area via the use of sterile insects. What happens here in a typical insect life cycle, the male and female mate with each other to produce a large number of fertilized eggs. Okay, and eggs grow through a series of developmental stages to produce a large number of offspring to continue the life cycle. Okay, now in this approach to reduce their population, male insects are actually exposed to some kind of radiation or other treatment in the laboratory to render them infertile. And then these sterile male insects are spread over the infested area. Okay, and in the field, the females mate with these sterile males, but definitely no offspring will be produced, and hence there will be a control on the population. So over the course of time, the insect population will be reduced. And the example of this approach is eradication of boll weevil, an insect responsible for the loss of cotton crop in USA. We did see that some $1 million were actually lost. Like some $1 million loss was there to the economy, US economy back then due to this particular pest. Okay. So the answer will be eradication of boll weevil was achieved by the use of option A, sterile males. Okay. Now, the next question is a superior breed of female is known as. Now, in animal biotech, superior female breeds refer to those that have some desirable characteristics like high fertility, disease resistance, improved productivity like milk, meat, eggs, or wool, and genetic qualities beneficial for selective breeding programs. Now, these superior breeds are often targeted for advanced reproductive technologies, genetic engineering, and other biotechnological applications. Now, there are some general terms in animal biotech. One is brood stock, which is commonly used across species to refer to high quality female and male animals used for breeding, particularly in aquaculture, poultry, and livestock. Then there's the term foundation female, which, as the name suggests, is a female whose offspring forms the basis from the genetic basis of a breeding program and which is applicable across many species. An elite breeder refers to the females in the top tier breeding programs, often used for animals with exceptional reproductive performance and genetic traits. Now, in case of cattle or bovines, the term dams is a term often used for superior female cattle, particularly in breeding programs, indicating like which is dams are often able to produce high milk and can show strong mothering traits. Then there are elite cows. These are genetically superior cows used in artificial insemination or embryo transfer programs. Then there's foundation cow, a superior cow whose offspring are expected to serve as a genetic base for a herd. Then in terms of sheep or ovine, ewe is a female sheep, and when superior, they are often referred to as foundation ewes or elite ewes, particularly in breeding programs for wool or meat production. Then in case of goats, doe or nanny goat is a female goat. Superior goats for dairy production are often called elite does in selective breeding or dairy programs. In case of pigs, sow, a breeding female pig, superior sows with high fertility and good litter performance are again referred to as foundation sows or elite sows. And in case of horses, it's a mare, which is a female horse, and a superior breeding mare with exceptional genetic qualities is often termed as a brood mare or a foundation mare. Then there's something called elite mare as well, used specifically for mares or female horses with high performance in racing or sports. These are actually they are actually valued for possessing superior traits and as well as passing it on over to the offspring. In case of poultry like chickens, ducks, etc., there are layer hens, which are superior female chickens known for their high egg laying capacity. Some They are also known as foundation hens or elite layers. Uh, Breeding hens in breeding programs for both meat as well as egg production. Top hens are elite breeding hens. In case of fishes and shrimps, aquaculture, there are broodstock females, which are superior female fishes or shrimps used in hatcheries for producing high quality offspring. And in species like Atlantic salmon or tilapia, the term elite broodstock is used for genetically superior females. Then there's camelids, which include camels, alpacas, llamas. Again, the term dam is used for female camels, dromedaries or Bactrian or alpacas in breeding programs. Superior females can also be called as elite dams. In case of rabbits, doe is a female rabbit, and superior ones are called elite does or foundation does. It can be used for breeding, for meat, for fur. And then in case of buffalo, you have dairy buffalo, which are superior female buffaloes, particularly with high milk yielding capacity, like the Murrah breed, are often called the elite buffalo cows in dairy improvement programs. So, a superior breed of female is known as option B, dam. Okay. Now, the Bt toxin is an insecticidal toxin obtained from which particular species? So, you might be knowing it very well. So, a genetically altered crop is produced to develop resistance against this crop feeding insects. And one of the approaches is to genetically modify the plant, which actually expresses a toxin that can kill this particular insect, which is feeding on it. But it will be safe for human consumption. So, Bacillus thuringiensis is the bacterium which secretes an insecticidal toxin, and spraying this Bt toxin was in circulation to control the insect population. And when transgenic plants were produced, which expresses Bt toxin in their somatic cells, and when this insect or feeds on the plant, the toxin can actually kill the insect feeding on it, basically by causing internal bleeding. Okay, so the correct option will be this toxin is obtained from which particular bacterium? It's Bacillus thuringiensis, option B. Now, glyphosate interferes with the biosynthesis of which particular amino acid? So, again, this is used in creating herbicide resistant plants. What happens here is glyphosate is one of the first herbicide which was designed to kill weeds. And what it does is it interferes with the biosynthesis of aromatic amino acids like tyrosine, phenylalanine, and tryptophan. What it does is it inhibits this particular enzyme EPSP. Okay, this one, 5-enol pyruvyl shikimate phosphate. The enzyme catalyzes the this APSP enzyme catalyzes the conversion of shikimate 3 phosphate to 5-enol pyruvyl shikimate 3 phosphate. Now, what happens here is the treated plant isn't able to produce these amino acids as well as the protein that it needs, and hence it dies. So there are two approaches adopted to develop herbicide resistance in crop plant. First is the genetically modified crop plant is designed with an alternate pathway to supply the aromatic amino acid to compensate the inhibition of EPSP, because the plants might need it in some other biosynthetic pathway or some other metabolic pathway. And few bacterial strains actually use an alternate form of EPSP that is resistant to glyphosate inhibition. So the modified version of EPSP gene was isolated from Agrobacterium strain CP4 and cloned into the crop plant to provide herbicide resistance. Okay. Um, and this crop plant, this herbicide resistant crop plant is actually, so far this crop plant is available in with like soy, maize, sorghum, canola, and cotton. So glyphosate actually interferes with the biosynthesis of aromatic amino acids, which is option C. Sorry, option C in this case, tyrosine. Okay. The glyphosate herbicide is an inhibitor of which particular enzyme? We just now looked at it. It will be option C, EPSP synthase. Now, Bell Boy F1 is a pepper variety that is resistant to which particular viral species here? So, now these are different kinds of viral species or insects or worms, and the variety that have been developed by genetic engineering. These are the examples of it. So, the question asked is about Bell Boy F1. This is the paper here, Bellboy F1, which is resistant against mosaic virus. Okay, so the option will be option D, mosaic virus. Okay, now a totipotent part of the plant that can be used to generate the whole plant is called what? What is the term used for it? Now, few things. Uh, there can be different parts of the plant which can be used to generate the whole plant. Okay, now suppose this is the root of the carrot plant, right? And you cut it out into small fragments, and these fragments can actually be cultured in an appropriate nutrient medium. Then from those fragments, there are free cells in the suspension, like cell culture medium, which begin to divide, and thereby forming an embryoid, which is a somatic embryo, which develops from these cultured free cells. Okay, and then the this particular like cell-free suspension, there is this embryoid developing, and when the plantlet cultured on agar medium is first actually cultured on an agar medium, like solid nutrient medium, and then when it grows into a plantlet, this is transferred to the soil to let it grow into an adult plant. Okay, so from certain parts of the plant, one can get a complete adult plant. Okay, one can culture it properly under appropriate nutrient conditions as well as environment conditions, so that it grows into a fully mature plant. Okay, and a plant explant, when we talk about the term explant, is a fragment of plant tissue that is used as a starting material for plant tissue culture. Now, examples of explants are like root fragments, which we saw in the earlier slide. It can be shoot fragments, it can be sections of leaves or petals, etc. And from there, the explant can be used for regeneration or non-regeneration techniques. Now, a plant callus or calli is a mass of undifferentiated cells that grow over an explant as a result of wounding or when growth factors are added to the culture media. Okay, a callus can give rise to shoot buds and somatic embryos that can form complete plants, or a plant callus can be transferred to a liquid medium and produce cells that are used to produce metabolites. So, if we were to answer the question, a totipotent part of the plant that can be used to generate the whole plant is called an explant. Okay, callus develops when there is some kind of injury to the cell or to the plant part, and then it develops on an explant mostly. Now, the standard temperature and pressure in a standard autoclave should be dash and dash. Now, very briefly, I think in first few, I think in one of the beginning tutorial sessions, we talked about autoclaving stuffs. So for sterilization, autoclaving is a wet sterilization technique, and there can be different sterilization techniques as well. Now, autoclaving is a machine. It is a physical method of sterilization which kills bacteria, viruses, and even spores present in the material put inside the vessel using steam under pressure. So we talked about autoclaving liquid culture, right? For culturing E. coli cells, etc. We have to autoclave the media. We have to autoclave the containers you will be using for different type, like culturing either a primary culture or secondary culture, or using the tips that you need to uh, pipette out cells or pipette out solutions, etc. For competent cells and all, everything has to be autoclaved. Okay, or rather wet sterilized. Now, autoclave sterilizes the materials by heating them up to a particular temperature for a certain amount of time. And the autoclave is also called a steam sterilizer that is commonly used in healthcare facilities and industries for different purposes. An autoclave is considered a more effective method of sterilization because it is based on moist heat sterilization. So this is how an autoclave looks like basically, or it can be larger also in laboratory, like experiment, like research laboratories. We have like really big autoclaves as well. Okay. Or the thing is mostly the overall concept comes from a pressure cooker. Even for day-to-day sterilization purposes, one can even use a pressure cooker for sterilizing media, for sterilizing cultures, etc. Okay, I mean culture medium, not cultures. Cells will die. So the principle, the autoclave works on the principle of moist heat sterilization, where steam under pressure is used to sterilize the material present inside the chamber. The material here in question can be glasswares, media, liquid media, or tips, pipettes. I mean, not pipettes, tips, and then there can be different kinds of, beakers, measuring cylinders, etc. Those stuffs can be sterilized. Then many times during, suppose when you really want to work with RNA or ribosomes, those are very um, prone to degradation, right? So any kind of solutions you want to uh, prepare for your assays, or suppose for competent cells preparation, you need salts of divalent ions, right? So all those salt solutions are also autoclaved. So any kind of material, these materials can range from solutions to glasswares to cultures, etc. The high pressure increases the boiling point of water and thus helps achieve a higher temperature for sterilization. So water usually boils at 100 degrees Celsius under normal atmospheric pressure. However, the boiling point of water increases if the pressure is to be increased. Similarly, the high pressure also facilitates the rapid penetration of heat into deeper parts of the material, and moisture present in the steam causes the coagulation of proteins, causing an irreversible loss of function and activity of microbes. This principle is employed in an autoclave where the water boils at 121 degrees Celsius at a pressure of 15 psi or 775 millimeters of mercury. So, to answer this question, the answer will be, what is the temperature and pressure of a standard autoclave? The answer is option C, 121 degrees Celsius and 15 pascals pressure. Now, we are talking about sterilization, but there can be many different ways of um, like there are many terms that needs to be taken care of, like sterilization. When we are talking about sterilization, it is a process designed to eliminate all microbes, including both vegetative cells and spores, from an object, surface, or medium. This ensures complete microbial eradication, rendering the object or surface sterile and free of all forms of life. And the techniques can include heat, chemicals, irradiation, high pressure, and filtration. Okay, we talked about filter sterilization using uh, membranes with like zero point 0.2 micron pore size or 0.45 micron pore sizes, etc. Right? So it can be filter sterilization, high pressure sterilization, what we see in autoclaves, right? Heat as well as heat. This is a moist sterilization, and there's dry sterilization as well. Okay, in ovens, etc. Like at high temperatures. And there's disinfection, which involves the destruction of pathogenic microbes that can cause infection, though it does not necessarily kill all the spores. The aim is to reduce the number of microbes to levels that are not really harmful to health. While disinfection significantly decreases the microbial presence, it does not really achieve the complete microbial eradication associated with sterilization. Then there's antiseptics, which are chemical agents applied to living tissues to prevent infection by inhibiting the growth of microbes. These substances are used to prevent infection and control microbial growth on skin or mucous membranes. Antiseptics must be safe for application on living tissues and effective against a range of microbes. There's something called asepsis. Asepsis refers to techniques and practices aimed at preventing infection in uninfected tissues or environment. The goal is to avoid the introduction of pathogens into sterile areas or materials, and the techniques can include sterilization procedures, maintaining sterile fields, sterile areas for work, using protective barriers to prevent contamination. Now, these are some of the physical methods of sterilization, like sunlight, which actually utilizes UV rays for germicidal effects. This reduces microbes in natural bodies of water as well. The key agent is UV rays, and this can be used for water sterilization. There can be heat sterilization, which involves dry heat or moist heat to kill the microbes through protein denaturation and coagulation. The key agents are either dry heat or moist heat, sterilization of glassware, surgical instruments, chemicals, etc. Dry heat, like both of these come under this only. Mainly dry heat uses high temperatures to induce oxidative damage and protein denaturation, like red heat, flaming, incineration, hot air oven, etc. Flaming is many times when you are using laminar hood, right? So you need to use a spreader. You need to many times after you autoclave the stuffs, like autoclave your culture, your media. Many times there are conical flasks with liquid culture inside. Okay, so the neck or mouth of the flask is actually plugged with a cotton. Now, it might happen that while you're handling, you have to definitely remove the plug to pour your media. Like, suppose you need only 10 ml of media and you have like 100 ml of media autoclaved. So, you actually pour it, or suppose you need to add something into that liquid medium. Then, what happens is you have to remove the cotton plug, right? So, it has to be done carefully in inside an hood and under, like you have to kind of show the neck to the flame, so that you this is also kind of a dry heat sterilization method, so that due to your handling you don't really contaminate it with any microbe. Okay, so all these things have to be, you have to be, the user or the person in the laboratory has to be careful while doing these bacterial work or any microbial work, okay, to prevent contamination. So this is what refers to, like this is what flaming means. Mostly, like, or suppose we were talking, you guys might be aware about spreading the cells on an agar plate, right? So there are glass spreaders as well, glass spreaders, or you know about streaking, right? So there are loops, streaking loops for bacterial colonies or cultures. Okay, so what happens in there, you actually show that metallic loop or the glass spreader onto a flame, heat, sterilize it, let it cool down, and then you use it for your experimental purpose. Okay, so that is what flaming refers to. Incineration, hot air oven, basically for sterilization of metal and glass instruments that I was talking about, or chemicals as well. Then moist heat uses steam to kill microbes through protein coagulation. Uh, you can do boiling, you can pasteurize it, inspissation, vaccine bath, autoclaving. Basically, you are sterilizing the media, vaccines, and heat sensitive materials. One can also use ozone gas for disinfection produced from oxygen through electrical discharges, and which is used for sterilization of medical and laboratory equipment. You can use filters to remove microbes from liquids or gases. There are candle filters, right, in your these water purifiers also uses candle filters, etc. There are different kinds of syringe filters that I was talking about earlier in laboratory purposes. Air filters are there, like when like in laminar hood, or in a laminar hood, or any kind of different kinds of hood in chemical laboratories. All these have different positions or different ways to filter out airs. I mean, filter out particulate matter from the air surrounding you. So there are air filters, there are membrane filters, there are asbestos disc filters, basically for sterilization of heat sensitive liquids and air. You can also use radiation, which utilizes ionizing and non-ionizing radiation to kill microbes by damaging the DNA, like gamma rays, X-rays, UV radiation. These are highly lethal, especially X-rays, gamma rays. UV radiation is also lethal. sterilization of disposable items, surfaces, and environments. And then there are few chemical methods of sterilization like alcohols, um, denature proteins of microbes, ethyl alcohol, even like for rupturing the cells, we see like for DNA isolation or plasmid isolation or RNA isolation, we did see that we use a combination of chloroform, alcohol, right? So alcohol is actually a denaturing agent as well, right? Also, we saw in the phase inversion, like phenol chloroform uh, isolation, phenol chloroform method for isolating nucleic acids. We see that phenol is actually added to denature the proteins, right? So alcohols, denaturation of proteins of microbes. Agents can be isopropanol, ethyl alcohol, methanol. Applications, skin antiseptics, disinfection of surfaces and equipment. You can use aldehydes, which can also be bactericidal, sporicidal, and virucidal agents. Formaldehyde, glutaraldehyde, orthophthaldehyde. Sterilization of medical instruments, vaccines, and laboratory equipment. Phenols disrupt cell membranes of microbes. Phenols, chrysols, chlorhexidine, chloroxynol, hexachlorophene. These are used to disinfect surfaces, wounds, and medical instruments. One can even use halogens, which are oxidative agents used to kill microbes like chlorine, iodine. Disinfection of water. We know that like swimming pools or in general drinking water facilities usually use chlorine or iodine for disinfection of the like in water treatment bodies, as well as in skin one uses iodine tincture as well. Oxidizing agents releases oxygen radicals to kill microbes like H2O2, peracetic acid, which sterilizes surfaces, equipment, and some medical materials. One can even use salts like heavy metal salts to coagulate proteins like copper salts, mercury salts, silver salts. Disinfection and preservation of sera and other materials. Dyes can also be used, which are actually bacteriostatic agents. Not really, they won't be killing the bacteria, but it can be used for skin and wound antiseptics like aniline dyes, acridine dyes. And then there are vapor phase disinfectants like which use uh, gaseous agents to sterilize materials like ethylene oxide, beta propylolactone, which are used for sterilizing heat sensitive materials and inactivation of vaccines as well. Okay. Now, which of the following is a minor salt in MS media? MS media is something which is used mostly for plant cultures. Okay. And so the overall MS media is having a like carbohydrate source, glucose. There are major salts which are really essential. There's like um, source for iron, salts are added, and then there are minor salts. So major salts are like really essential, required in larger quantities for plant growth, while minor salts, or you might have read about micronutrients, which are needed in trace amounts but are actually equally important for plant metabolic processes. And both types of salts are actually crucial components of plant culture media. So MS media, which we are talking about, is Morashik and Skoog medium, one of the most commonly used media in plant tissue culture. It contains both major and minor salts in concentrations optimized for plant cell growth and regeneration. So major salts in MS include ammonium nitrate and potassium nitrate, and minor salts include manganese chloride, zinc sulfate, and copper sulfate. Okay. So the answer out of these options will be copper sulfate. Okay. A surface sterilization of an explant is usually achieved by treating it with which of the following substances? So, suppose you have used leaf as the part from where you want to generate the whole plant. Okay, so first you have to sterilize it, but before sterilizing it, you have to sterilize the surface on which you are going to work. Okay, so selection and surface sterilization of the explant, and so this is actually done using this detergent tween 20, and then again washed with sterile distilled water. So the you are getting the answer from this particular picture only. The option will be option B, tween 20. So what is done next is sterilization using 70% ethanol. Now you guys must have, if you have worked in laboratories or for your practicals, you might have seen the usage of 70% crude ethanol a lot. Okay, why 70%? Why not 100%? So basically what happens here is uh, mostly in 100% ethanol, 70, if you are diluting it a little, uh, the surface tension is too high, and the ethanol, like 100% pure ethanol, crude ethanol, can't really penetrate the cell surfaces or cells, like like microbes contaminating your experimental bench or your experimental materials. Okay, so 70 per diluting it a little actually helps it in penetrating inside. Okay, and then it's washed using sterile distilled water, and then again there's mercurous chloride, HgCl2, and then again washed with distilled water. So this is the overall sterilization of the explant. These are the techniques used, but for answering, uh, the answer for this question will be option B, tween 20. Surface sterilization of an explant is usually achieved by treating it with this detergent. Okay, now which of the following is a disadvantage of artificial insemination? What happens in artificial insemination is it involves the introduction of the male sperm into the reproductive tract of the female animal artificially, and the availability of superior breed animal is due to the artificial insemination. What happens is the semen is collected from the superior breed male in a test tube, and the quality of the sperm, like motility and the number of sperms in that particular semen, like in that particular test tube, whatever sample has been added, the number is checked through a, like under a microscope, and then it is mixed with extenders like milk, yolk, glycerine, and antibiotics, and stored in liquid nitrogen for future use, right? And before artificially introducing into the reproductive tract of the female, what happens here is um, one has to be wary of the estrous cycle of the female, like as to when she is going to ovulate. Okay, and then during the time of ovulation the semen is injected um, in the female reproductive tract to facilitate the process of fertilization, and mostly it's done under skilled technicians hands. But what actually is a disadvantage here is there are several advantages definitely as compared to the natural breeding. Like the male of a hybrid, commonly known as sires, is very costly in comparison to the semen from them. Definitely, and through a natural breeding process, many diseases can be transmitted to the female via mating or during mating. These possibilities are much reduced by using artificial insemination procedure. The hybrid animal imported from other countries need to go through quarantine process to ensure no spreading of disease, and this process is like really costly and time consuming. But there can be disadvantages as well: loss of natural traits and reduction in disease resistant traits. Okay, so out of these options, the correct option regarding the disadvantage of artificial insemination is loss of natural traits. Okay, you don't really need to procure sires, you need to have the semen from the hybriding male. Okay, and you don't really have an increase in disease resistant traits, it actually decreases. That is another disadvantage of artificial insemination. So the answer will be option C, loss of natural traits. Okay, which of the following recombinant protein is used for the production of cheese? So recombinant protein used is chymosin, which is a proteolytic enzyme, LYTC, lytic enzyme obtained from calf stomachs, which is used to coagulate milk during cheese production. Okay. So the correct option is option A, chymosin. Now, in monoclonal antibody production, the dash and dash cells are fused. Before going into this question, let's look at what is a monoclonal antibody and what is a polyclonal antibody. So in monoclonal antibody, the thing is the antibodies are specific. These antibodies recognize a specific type of antigen, but in polyclonal antibodies, multiple parts of the same antigen is recognized by the set of antibodies, polyclonal antibodies. Okay. So in monoclonal antibodies, antibodies are used in the adjuvant therapy to remove the infectious agents or toxic substances, and these are actually generated by hybridoma technology. What happens here is myeloma cells, the like proliferating capacity of myeloma cells are used, and the immunizing capacity of B cells are used, and then these are actually fused to produce monoclonal antibodies. Okay, so myeloma cells are cancerous or tumor cells which are derived from plasma cells, a type of white blood cell responsible for producing antibodies or immunoglobulins. Now, what happens in hybridoma technology? Myeloma cells, which can proliferate, right, are fused with the spleen cells, from where we can get B cells from a mouse that has been immunized with a specific antigen. Now, the resulting hybridoma cells combine the immortality of myeloma cells or the tumor cells with the antibody producing ability of the B cells, allowing for the production of large quantities of the specific monoclonal antibodies. And how do you select it? Once the hybrid cells are created, they are selected for those that produce the desired antibody and can be cultured indefinitely. Okay. Now, when we talk about monoclonal and polyclonal antibodies, so monoclonal antibodies are specific to a single antigenic site. For example, there's this antibody rituximab, which targets the CD20, only this part on the B cells. But polyclonal antibodies are a mixture of um, like antibodies targeting multiple epitopes of a virus or of the particular antigen in question. Okay. Um, then monoclonal antibodies are actually homogeneous antibodies from a single B cell clone, but polyclonal antibodies are a mixture of diverse immunoglobulins from multiple B cell clones. Monoclonal antibodies are produced from one plasma B cell clone, whereas polyclonal ones are produced from different plasma B cell clones. And production of monoclonal antibodies are actually expensive as compared to the polyclonal ones, because monoclonal have to be really specific and it takes a lot of time to generate, or production of monoclonal antibodies is costly as well as time consuming, but polyclonal antibodies are faster to produce, and monoclonal ones are highly specific, but polyclonal antibodies are less specific and have a broader uh, detection range. Okay. Hybridoma cell lines, you require it in case of monoclonal antibodies, but in case of polyclonal antibodies, you don't really need it. So monoclonal antibodies can be used for therapeutic drugs, and polyclonal ones can be used for general research and applications. So now the answer to this question, in case of monoclonal antibodies, this is about the hybridoma technology. So which kind of cells are fused? Myeloma and B cells, option A. Okay. Now in hemophilia, which of the following component is in deficit? So in hemophilia, factor 9, factor 8 are in deficit, which um, leads to blood clotting defects. Okay. So the and also even one uses recombinant clotting factor 8. In a normal individual, blood loss from a damaged blood cell is prevented by the formation of a blood clot. And blood clotting is a series of a cascade process involving different factors. And factor 8 is deficient in bleeding disorders such as hemophilia. And recombinant factor 8 is supplied to improve the condition. Okay. So the correct option is option C, factor 8. Okay. So this is it for today's session. I will stop my screen. Thank you for joining. Okay, we'll meet again um, in the on next Saturday, and I will also have an extra session where I will try to take up questions uh, from all the from the complete course content other than the ones which we discussed here in all these tutorial sessions, and try to discuss those as well. Okay. All right then. Thank you for joining. See you on Saturday. Okay. Next Saturday. Yeah. Thank you. I will end the meeting for all of you. Yeah. Thank you, Gracilia. Thank you, Dashisha. Yeah. Thanks, Jessica.